I'm Lucie. I'm a postdoctoral research associate in Manaf Botanical Lab at the University of Cambridge. And so our lab main goal is to unravel the biology of pediatric brain tumor and a specific subtype of pediatric brain tumor, which is pediatric high-grade glioma that will, of course, introduced in introduction. And so as academics, we really want to understand how this tumor grow, maintain, and that is essential for finding relevant therapeutic targets. So brain and central nervous system tumors are the most frequent cancer in children. And pediatric high-grade glioma, so the subtype that we are focusing in our lab, accounts for 10% of pediatric brain tumors. So although they are rare tumors in children, they're causing more death than any other childhood malignancy, where only 10% of the patients survive 5 years post-diagnosis. And histone mutant pediatric high-grade glioma can fall in two main categories. So diffuse midline glioma, DMG, diffuse hemispheric glioma, DHD. And they are highly heterogeneous tumor entity driven by clonal mutation and are often associated with a range of genetic alteration. So as I mentioned, different histone mutation correlates with different brain regions, ages of onset, and survival outcome. So for the DMG, so the diffuse midline glioma, it exclusively occurs in midline locations, so namely thalamus, brainstem, and spinal cord, with area with restricted surgical accessibility as they carry most of your automated behavior function, like breathing, for example. It has age of diagnosis between 6 years and a half and 10 years old, and the survival post-diagnosis is between 10 and 13 and a half months post-diagnosis. While for DHD, so the hemispheric glioma, it's confined to the cerebral hemisphere with a median age of diagnosis of 13 years old and a median survival post-diagnosis of 18 months. So interestingly, the anatomical distribution of this tumor is associated with distinct histone mutations. So for the DMGs, it's associated with H3.1 and H3.3 K27M mutation, while DHG is associated with H3.3 G34R mutation. And you also have some wild-type histone pediatric high-grade glioma, but in the lab, we are only focusing on the histone mutant pediatric high-grade glioma that represent 50% of the patient. And among this 50%, 80% of them are DMG patients and 20% are DHG. So, as I just told you, they are driven by different oncogenic mechanisms, but they're also different from different developmental origin. So for the DMG, so the one carrying H3 K27M mutation, they likely originate from oligodendroglial progenitors within the midbrain and the hindbrain, while for the H3.3 G34 mutant, so the DHG, hemispheric glioma, they likely they derive from forebrain interneuron progenitors in the lateral ganglionic eminence. So, histone mutation also co-segregate with distinct partner alteration that are highly heterogeneous, making this tumor heterogeneous on a molecular and genetic level, both inter- and intratumorally, making the treatment even more complicated. So for DHD, neurosurgery and DNA damaging agents do not improve survival, while for DMG, there's no surgery possible, as I told you, due to the location in critical midline structure. So there is a huge lack for targeted therapies. And in that case, it's very important to consider that the brain at the age of onset of pediatric high-grade glioma, the brain is still under development. So it's very important to consider that. So different treatments could have an impact on structure and function of the brain. And most of the treatment that I used nowadays for treating pediatric high-grade glioma, even if, as you saw, the survival outcome doesn't really change, are mainly treatment that have been developed for glioblastoma in adults, while we perfectly know that it's very different molecular processes in between the two diseases. So we really need a histone mutant model, or different histone mutant models, because as you can see, there's many different combinations, reflecting this anatomic and genetic heterogeneity of the disease. So that's where our lab steps in. So before I even joined the lab, Manav, our PI, Michael, who was a research assistant at the time and is now a PhD student, and Antonella, the senior postdoc of the lab, they managed to create 16 different models of this histone mutant glioma, and 12 of them are H3K27M mutant, so DMG, midline glioma. And so how did they manage to do that? It was really hard work, as you can imagine. And so they carefully designed different vectors, either piggyback or CRISPR vector, carrying different mutation. And they chose the mutation based on what was the most frequently seen in patients. And so they delivered the combination of the different vectors into discrete neural progenitor niche using in utero electroporation. After that, the embryo were returned back to the uterus of the mother. The pregnancy was carried through delivery, and then the mice developed and grew. And so they developed tumor, and at some point they developed neurological symptoms from the presence of a big tumor taking up the brain and requiring euthanasia. So at that time, then we reached the human endpoint of the study, and we euthanized the mice, allowing us to harvest some tissue. So they were able to thoroughly characterize this tumor on both histological level and molecular biology level, confirming that there were models that really were reflecting discrete tumor subtype that we find in patients with these different mutations. But while in utero electroporation technique are highly relevant for translation to human models, some of this model can take up to 2.5 years to develop neurological symptoms. So as you can imagine, there was a need for a faster alternative, creating a tool to answer any biological question or for rapid drug screening, for example. So from that angle, they were able to derive ex vivo cell lines from the different tumors. And these ex vivo cell lines were able to engraft in immunocompetent mice with high penetrance for most of our models. And so now we have the perfect tool to answer any biological question that we would like to answer. And so at the moment, now we have 3 different projects, main project going on in the lab. So of course, I'm mainly going to focus on my project, but I'm still going to talk to you briefly about the project of Antonella, the senior postdoc of the lab, and Michael Days now. He started his PhD more than a year ago now. So both of the projects are focusing on unraveling drug-resistant mechanisms. So for Antonella's project, she wants to identify resistant mechanisms in new therapeutic targets. So for that, she managed to generate resistant clone on which it's currently undergoing for the analysis. She did exome sequencing to identify acquired mutation, and she also wants to perform RNA sequencing to analyze the transcription profile of this resistant clone. As for Michael, his project is mainly focusing on genetic barcoding of these syngeneic models. So this is still currently undergoing. And then he will engraft these barcoded models in immunocompetent mice, as you can imagine. And then he will apply either treatment, radiotherapy, or combination of both, of course, with control. And then he wants to perform single-cell sequencing to identify clonality and resistance signatures in the different models. As for my project, I'm mainly focusing on the tumor immune microenvironment in these different models. So I did a broad immunophenotyping of most of the models that we have using higher parametric flow cytometry. Of course, I will go a little bit more in details later on. And I will also like to do single-cell sequencing to better characterize the tumor infiltrating immune cells and to better understand the interaction with tumor cells or with other cells within the environment that are neither immune or tumor cells. And of course, all of this to be able to find new therapeutic targets, because if we can understand how which kind of cells are present, are they beneficial or detrimental for the tumor, we can find a way to act on different angles on these different models. But before getting into the details of my research project, I want to give you an overview of the unique properties of the central nervous system, especially regarding the immune surveillance of the central nervous system, because it had long been thought to be kind of, we call it immunoprivileged, but it's not really immunoprivileged, it's more has unique properties, but you still have immune surveillance within the central nervous system. So at least we're all on the same basis for understanding a bit the results that I got. So the main unique, not phenotype, but part of the central nervous system is the presence of the lymphatic system. So the lymphatic system is for fluid exchange, responsible for waste clearance and central nervous system immune surveillance. Why is it unique? Because the CNS is actually lacking a typical lymphatic system that you probably know better than the glyphatic system. So you don't have any lymphatic system within there. It's a little bit more complicated. So I'm going to explain you why. So fluid and immune cells can traffic in both directions through the perivascular space surrounding blood vessels, but also through the cerebrospinal fluids that is present around the brain as well as in the brain ventricle. And around the brain, it communicates with the only lymphatic vessels of the central nervous system, which is within the dura mater. The dura mater is part of the meninges that is composed of 3 different layers. So dura mater, you have the only lymphatic vessels of the central nervous system. Then you have the arachnoid mater in between, and the innermost layer that is directly against the brain parenchyma, the pia mater. And so the CSF, so the cerebrospinal fluid, is secreted by the choroid plexus, another entry point for immune cells, and these immune cells can then migrate to cervical lymph nodes. So you're probably more familiar with what happens usually in normal organs. So you have immune cells extravasating from the blood vessels. They will go in the tissue, do their job, and then most of the time they will go within the lymphatic vessels, and then they will go to the lymph node, the closest lymph node of the organ, depending, and presenting antigen-presenting cells. So to then trigger, for example, a lymphocyte response, and then they will go back to the, so it's way easier. It go out and then it goes in the other one, go to the lymph node. While here, because the brain is a bit specific, because the blood vessels are also a little bit specific because of the presence of the blood-brain barrier that I will explain to you, the cells cannot just go out of the blood vessels. It's a little bit more complicated. So it needs specific condition to be met for them to be able to extravasate. And then they go in the perivascular space, and then within the CSF, and then it's drained to the lymph node. And so all of this anatomical niche represent anatomical site, represent immunological niche through which the immune cells can traffic both in and out. And so I mentioned the blood-brain barrier. So the blood-brain barrier is the fact that your endothelial cells composing your blood vessels have tight junction, preventing leakiness, that are really tighter than the one you have in normal tissue. So meaning that even small molecular weight protein most of the time cannot go out of the blood vessels. And then you have what I mentioned to you, the perivascular space that is acting kind of like a tampon space. And then on top of that, you have the astrocytic end fit as another layer of protection before the brain parenchyma. And so you might wonder why it's so important to actually kind of dampen the inflammation within the central nervous system, because actually when you have inflammation, it's very detrimental for neural processes and cognitive function. A great example is when you're sick, for example, you have a cold or the flu, COVID, I don't know. Most of the time you will experience what we call sickness behavior that have symptoms that are kind of similar to depression. So fatigue, loss of appetite, you can have change if you sleep schedule, etc., etc. So it's a great example of how inflammation can actually impact greatly your immune function. That's why it's kind of trying by the system to dampen a little bit the inflammation. Of course, it's not perfect, but it dampens a little bit. And then another addition to that is that most of the time when you have inflammation, you will have swelling of your tissues. And because of the anatomical location of the brain within the skull and the spine within the vertebra, it has an anatomical limitation of the swelling of the tissue. So if it swells too much, then you will have an increase of intracranial pressure, and that is also very detrimental for your neural processes and your cognitive function. And so what kind of immune cells can you find at steady state in the central nervous system? Well, so here it's another representation. You find back the dura mater with the only lymphatic vessels of the central nervous system. Here you have the arachnoid mater with the cerebrospinal fluid that is going through. And here you have the perivascular space that I told you about, with again your astrocytic enphyte that are surrounding the brain parenchyma, adding another layer. So in normal condition, within the brain parenchyma, you only find microglia, which are the resident macrophages of the central nervous system, and a few monocytes and lymphocytes that acts as an immune surveillance system of the central nervous system. But in inflammatory condition, such as pediatric high-grade glioma, for example, you have the blood-brain barrier that can become more leaky, and therefore you have a higher infiltration, or you can have a higher infiltration of a broader type of immune cells within the brain parenchyma, a lymphocyte, neutrophils, monocyte, etc., etc. And so now the tight junction that were forbidding the cells from extravasating from the blood vessels can now go out, and same way for some proteins or chemokines that were not always able to go through because of the molecular weight. So if the blood-brain barrier is not intact anymore, then it can go through and gain access to the brain parenchyma. So, sorry, now that we are all on the same basis on how the central nervous system is a bit more specific compared to other organs regarding the immune surveillance, I want to go back to my project because it's, as you can imagine, mainly immunofocus. So for my research project, I really want to focus on the tumor immune microenvironment. And for that, I want to do a global central nervous system immunophenotyping in the context of pediatric high-grade glioma, of course. And I want to answer three main questions. Are histone mutation and different partner alteration influencing the tumor immune microenvironment? Does the tumor affect the global immune state of tumor-bearing brain? And are tumor triggering immune cell recruitment through the meninges? So for that, I investigated the tumor immune microenvironment composition of 10 different models. But today, as you can imagine, for time restriction issue, I'm only going to talk to you about 5 models, on data that I obtained for 5 models that are carrying H3K27M mutations, so they are diffuse midline glioma. So how did I do that? For my experimental design, I did it with the help of the research assistant of the lab, Carmo, especially for cell culture. So we implanted the different cell lines in our immunocompetent mice, as you can imagine, because we're studying the immune system. And then I harvested different tissue at a specific time point based on the survival data that was obtained by Michael and Antonella before I joined the lab. So I took the median survival that they obtained for the cohort that they did, and I called the animal at that specific date because otherwise it would have been possible for me to do that experiment if I waited for the human endpoint to be reached, because it can be at any point during the day. And unfortunately, this kind of experiment are a very long day. So if you start at 3, then you will finish, I don't even know, at 4 in the morning. So it's not possible. So we did the best we could do for it to be feasible, humanly possible. So I harvested 4 different types of tissue. So the distal parenchyma, so basically the brain without the tumor, then the tumor and the proximal peritumor parenchyma, the calvarium dura mater, so the dura mater only that is only on the skull, on the top part of the skull, and the pia mater all around the brain, and on which I perform multiparametric flow cytometry with 27 color panel. So for those that are familiar with flow cytometry, I won't go into the complex gating strategy, but if you want to ask me, feel free after the talk. Today I will only present you 5 different models, KP, KPP, KPF, KF, and KN. So all of the data that I'm going to show you now are unpublished data. So depending on what you want to do with them, please do not share, please. So they are all carrying the H3.3 KN27M mutation, and then additional mutation. So some of the model are acting kind of our control for the other model. So for example, when we compare KPP to KP, because they are both of them carrying the P53 loss of function mutation, it's allowing us to understand what the addition of PDGFR mutation does in this context. And in the same way, we can compare, for example, KPF and KF, that both carry FGFR mutation. So when you add P53, it allows us to understand what the addition of P53 in the FGFR context does here. So we find back our model, KP, KPP, KPF, KF, and KN, with the survival data that was obtained before. I call the animal on the median as survival. So for example, 22 days per surgery for KP, 12 days for KPP, 33 days for KPF, 50 days for KF, etc., etc. And you are going to notice that I also have the sham-operated animals that are our internal control. That's the best control we could have. So the sham-operated animal underwent exactly the same surgical procedure, except that instead of injecting tumor cells, we injected PBS, so salaric, within the ventricle, in the brainstem. And so here you can see that the proportion of the lymphoid versus the myeloid cells, and you can appreciate that in the FGFR cell as well as in KN, you have a higher infiltration of lymphoid cell within the tumor microenvironment. And if we go a little bit more in details in the different immune subtypes that we have in the tumor microenvironment, you can appreciate that the differences in the lymphoid infiltration within these 3 different models is mainly carried by a difference in the CD8 T cells infiltrating the tumor microenvironment. So from that, we can conclude that in the context of the FGFR1 mutation, CD8 cells are recruited to the tumor microenvironment as well as in the NF1 mutation context. And as I mentioned, when you compare KF and KPF, with the addition of P53 mutation, it seems to inhibit the lymphoid cells recruitment because you have a lower infiltration of CD8 T cells. Of course, at the moment, it's only correlated. We are going to investigate deeper into that. So our hypothesis from that first question that we answered is that in the FGFR mutation context, lymphoid cells, and more specifically CD8 T cells, are recruited in the tumor microenvironment, where they could exert anti-tumor function, because the fewer CD8 you have, the shorter survival is, and the more CD8 you have, the longer the survival is for our animal. Of course, now we are investigating that deeper because it's our hypothesis. As for my second question, does it affect the global immune state of the central nervous system to have a tumor within the central nervous system? Well, it was kind of surprising, but tumor immune response seems it does not affect the distal parenchyma, with almost no differences in between the model or with the sham-operated animal. And as for our third question, what about the immune response within the meninges, or maybe cells being recruited through the meninges? So we looked as well at the immune composition in the different meninges layers, so the PR and the dura mater, because the arachnoid is impossible to harvest. Most of the time, the PR, you get contamination from the arachnoid matter, so you call it the lymphomeninges. So the higher lymphocyte compartment you have in the tumor, it seems to correlate again with higher lymphocyte within the PR matter, as well as within the dura mater. So CD8 T cells proportion in the meninges seems to correlate with the CD8 proportion within the tumor microenvironment. So we hypothesized that they could be recruited through the meninges. But of course, again, it's only correlating now. So we want to dig a little bit deeper into it. So now I'm just going to briefly tell you what's next for us and what other biological questions we would like to answer. Because of course, it was a lot of work, and it's very interesting to see the differences in between the immune composition, depending on whether they carry this or this mutation. And now we want to investigate deeper. So most importantly, we want to understand if our data match that of human samples, because of course we want our research to be the most relevant possible for human patients. So that's currently undergoing with our collaborator at McGill University, Nada Jabado and Claudia Kleiman. And another thing that is undergoing at the moment, mice are being treated. Do these recruited CD8 T cells have a beneficial impact on survival? So at the moment, I'm neutralizing the CD8 T cell with the antibodies. It's currently undergoing, so I cannot really tell you the results yet, unfortunately. And of course, it opened even broader global question. So what is the biological function of these infiltrative CD8 T cells? Because of course, at the moment, we have no idea if they are beneficial or detrimental. The CD8 neutralizing antibody will help us to understand that. But I want to do a deeper immunophenotypic of the lymphocyte compartment that I couldn't do with my first panel, because you can only do so many antibodies in a panel. How are they recruited to the tumor microenvironment? Because as I mentioned to you, you don't have that much lymphocyte at steady state in the central nervous system. So we want to understand how they're getting in. Of course, it's not the only disease where you will have a higher CD8 infiltration. For example, when you have a strong inflammatory response, such as in multiple sclerosis, for example, you have high lymphocyte infiltration with the brain parenchyma. So it's not specific to pediatric hyperglioma, but we want to understand what is the specific in the pathway, how they are getting in. Because if let's say they are beneficial, and we understand how they get in, then we can find a way to maybe promote it in other histone mutants that don't have CD8 recruitment. How is the blood-brain barrier disrupted in the different models? Because again, it can help us understand how immune cells are circulating within the central nervous system. And are they being recruited through antigen-presenting cells within lymph nodes? So the more, let's say, typical way of recruiting lymphocytes, or directly from the calvarium bone marrow, because you have a bone marrow directly in the calvarium. And it was quite recently, they're actually maybe 5 years ago, they were able to identify a connection between the calvarium bone marrow and the meninges. So, and you can have antigen presenting sign directly at the meninges, and then recruitment directly through the calvarium bone marrow. So I'm done for today. So thank you to my team, because as you can imagine, no scientists can work alone. So we need a team to do all of this. It's a team effort, collaborators that we work closely with as well in Cambridge and in Canada, for our facilities, and you for attention if you have any question.
Thank you so much, Lucie. It was really, really lovely talk. I think we've all learned a lot about the techniques and also your data. So thank you for sharing this unpublished data as well. And it's nice to have some exclusive parts as well. Do we have any questions to Lucie? I'm sure we do.
Sorry, it was not too drug focused, but unfortunately, I'm more basic science.
It was very much focused on translational research. So that's one of the key things we're covering. So it was really excellent. Any questions? I can't see anything in the chat. I can't see anyone hands up. Alistair, I'm sure you've got some question there. Are you waiting for a student? There you go. Perfect.
Hi, Lucie. I was thinking again, and you might like to tell the students about this. How do you manage to find the time to fit all these different aspects of the work into your day? What's the secret?
Well, we carefully design the experiments, so that's a big part of it. As I said, we are a team, so I have help For example, all the cell culture part, I don't do it. I'm very lucky. It's Carmo that is doing it. So I just have to tell her the date of the surgery, and then everything is ready. And then we have great help from the BRU facility where our mice are, because of course they check on them every day. Then they send a health concern if they have been issues. So we have help. So I guess, yeah, the experimental design make it all.
Yeah.
Thank you. I've got a question, Lucie, as well, if I don't see any hands up. Just wanted to ask you about the, looking at the kind of animal work, do you differentiate between female and male? And obviously there was some sort of papers published a little while ago about specificity to CD8 plus cells and image mice. Can you comment on that, please? Thank you.
Yeah. Very good question. I did my PhD on sex differences in pain, so yeah.
Okay, very relevant.
Yeah, so our models, usually when we implant the cells that we derived ex vivo, we respect the sex of the animal that they were derived from. And so most of our midline glioma model, so carrying H3K27M mutation, are males. And the G34R, the hemispheric one, most of the one we have are females. But I actually did it not that long ago to check if it was working as well, because again, it's such a not immune privilege, but unique immune properties that we might not have a response against the sex chromosome within the brain parenchyma. So I actually try, but not in the K27M model, in the G34R model. So I try to implant it in male. It works very well. We had 4 mice out of 5 that developed tumors. It's working very well. And the analysis of the data, I harvested the tumor as well. I want to check the differences because, of course, I'm expecting differences because it's quite well known in the literature. I mean, in the cancer literature, I don't know it all, but at least in the immune literature, we have very different immune system and we respond very different to inflammation between male and female and women and men. So of course, he will be very interested to see because whatever we find in our K27M model in male might not be applicable to what we will find in females, especially in the CD8 response. Most of the time, of course, it's never as simple, but most of the time you have a higher lymphocyte response in males, and while females rely more on the myeloid compartment. But again, yeah, we need to investigate, and we will do in some of the model, of course.
Definitely, and there'll be a point that you'll be separating the data as well and looking more specifically. But with this kind of techniques, like in utero electroporation, you take whatever you've got in terms of data.
Exactly.
So
unfortunately, we cannot pick.